Okay, so it looks like I am rate limiting between you guys and lunch, so I'm going to try and keep it interesting and also try to make up a little bit of time. So as Farouz mentioned, today's talk is going to be on solid dispersions and crystallinity. I think a very relevant topic given the preponderance of solid dispersion products that are on the market and in development at the moment. So what is it that we want to go ahead and take away from today's session? Well, we're going to try and talk about crystallinity in the context of a control strategy built around three major pillars: manufacturability, stability, and bioavailability. What I'd like us to walk away from today, if we take nothing else away, is really some basics around how we think about failure modes within our solid dispersion products, have an appreciation not just for the product that we're creating and how we create it, but even the environment where we're doing the manufacturing and the environment where we're storing the product, because this is going to be particularly critical for solid dispersions. And then understanding how crystallinity impacts the overall pK performance. We tend to think of it as a very binary thing. Crystallinity is fatal to a dispersion, and what we'll see is that actually low levels of crystallinity may be acceptable in some solid dispersion formulations. So quick biography, contact, get to know the speaker. In terms of my role at Merck, I'm currently a distinguished scientist and actually oral product portfolio and capability lead, which is quite the mouthful. Basically, I'm the senior scientific lead for our oral products group. As Farouz mentioned, I received my PhD at the University of Texas, have a master's degree in chemical engineering from Columbia, and also a bachelor's degree from SUNY Buffalo. So basically, been to the hot areas, been to the cold areas. New Jersey is kind of my Goldilocks space. Obviously, when I'm not doing science at work, I like to spend a lot of my time publishing and presenting. Engaging with the external scientific community is super important to me, and so what I'd like folks to take away from this as well is that it's not just about the talk. Feel free to reach out to me via email if you have questions or other things that you want to bounce ideas off of me. Always happy to chat science. So kind of with that as the intro and out of the way, talking a little bit about solid dispersions. I think everybody at this stage in the pharmaceutical industry life cycle is pretty familiar with what a solid dispersion is. Essentially, you're taking and transforming a crystalline active ingredient into an amorphous material to leverage the benefit of the increased free energy to enhance the solubility of that molecule. And oftentimes what we see is that amorphous APIs on their own, they're not physically stable, and so they're going to revert back to their crystalline form. So we work to stabilize those through the incorporation of excipients into that material to reduce the tendency for crystallization. And we break it down into three general categories of material. We have a crystalline solid dispersion. Oftentimes we're seeing these types of materials being leveraged for controlled release application, either oral tablets or long-acting implant type of systems. We then have this nebulous gray area of an amorphous dispersion or an amorphous solid solution. But in general, what we're looking for is an attempt to leverage molecular level dispersion of the active ingredient within the carrier system. The reality of dispersions is that it's actually probably an element of scale of scrutiny here around these, right? Depending on how closely you look, you can find some degree of heterogeneity potentially in that dispersion. And we've seen that play out not just with the active ingredient, but also with the excipients. So you could see an image of an extruded. Let's see if I can get to it. Right. So a little bit of a reach, but so the extruded image in the upper left of those four panes is a clear glass coming out of the extruder, characteristic of what you would expect to see. When we go and actually look at this more closely under AFM, what we see is phase separation, and that's actually phase separation of the vitamin E TPGS. So, these systems, they can be quite complex, and depending on the technique that you're using, and another reference that we have, we see that X-ray diffraction patterns, largely indistinguishable from one another, actually lead to materials with different physical stability profiles. So again, it's going to be very complex to connect the characterization to the product performance. And then once we've made a dispersion, it doesn't just stop there. We're talking about manufacturing an amorphous dispersion drug product. So it's going to be a tablet most predominantly. Sometimes it can be a capsule, and that needs to provide not just the solubility advantage, but we need to be able to prepare it in a way that it can be mass produced. We need to have it break down into the elementary granules that we have. Those granules can further disintegrate down into primary particles and ultimately dissolve, supersaturating the environment in the GI tract, leading to absorption of the active ingredient. And so fundamentally, breaking this down as an engineer and thinking about it in terms of dimensionless numbers, we can define three basic dimensionless numbers. We have a dose number, which is reflective of the solubility advantage for the system. We can think about it as a dissolution number, which describes the rate at which the release occurs. And we can also think about it in the context of an absorption number, where the material is going to be absorbed as it's transiting the GI tract. But the dose number actually carries significant importance for us when we think about how to identify products that may need to leverage solid dispersion technology. So this figure here is a figure that was developed and presented by Craig McKelvey many years ago. It's actually reflective of the Merck oral product portfolio. And what you see along the x-axis is increasing dose number, and what we see along the y-axis is a bioavailability estimate. And so these negative deviations that we're seeing as we go out in terms of dose number are reflective of when you get to a critical dose number, you can no longer support that product with a conventional formulation. And by going ahead and leveraging amorphous dispersion technology to increase the solubility, we're able to bring the bioavailability estimates more in line with the linearity that we would have projected from the lower doses when we administer at higher doses. But this begs the question of what happens when you have low-level crystalline material in your solid dispersion. And there's been numerous reports around this, looking at dissolution performance, trying to assess what occurs. And across most active ingredients, you tend to see that as you increase crystalline content, you decrease the dissolution performance, which should, in theory, drive a negative impact to the oral bioavailability of the product. And we know that, at least in the most extreme cases of fully crystalline material, the performance will be different than what we have with an amorphous dispersion. And so we need to make sure that our amorphous dispersions are controlled to minimize the extent to which we see crystalline content in the drug product. So this drives us to think about creating a control strategy, which is going to be based on the three pillars that I had mentioned at the onset. We need to make sure that the manufacturability is there to provide the right overall manufacturing process, composition of matter to our product. We need to make sure that it's stable for the shelf life of the product. We also need to make sure that it has the right bioperformance. And we're going to talk through today how we at Merck think about developing control strategies for amorphous dispersions. So pulling it back to the two basic technologies that nearly every organization is going to use comes down to hot melt extrusion and spray drying, and some of the talks already in the last 2 days have addressed these technologies. But I like to think about both of these technologies, while very different, and we'll talk about the differences, actually come down to two basic steps. And this is an analogy that my colleague Mike Lowinger had actually developed. It's going to consist of a mixing step and a quenching step. In the case of extrusion, the mixing step is very challenging. You're going to be mixing your materials at elevated temperature to create a single phase solid dispersion before you then go through a quenching step. Whereas in the case of spray drying, you're going to do your mixing step in a common organic solvent, which is actually fairly easy to do in many cases. In some instances, there can be a little bit more complexity depending on the solubility profile of your components in the formulation, both active and excipients. But it's really the quenching step in spray drying that creates the most challenge, because not only are you going to lock in the overall homogeneity of that dispersion, but you actually lock in the particle attributes as well, which can have a role in terms of downstream performance. So talking through some of the theoretical elements that we like to think about with extrusion as it relates to an amorphous control strategy. How can we make an amorphous system, and then how can we ensure that after manufacturing it is actually still amorphous? We can break it down into phase diagrams that essentially describe what's going on in the extruder, where really what you're looking for is melting point depression, and you're coupling that with the rheological characteristics of your formulation. So another colleague at Merck, Greg Troop, had developed what we term the Troop classification system, where we categorize the melt extruded systems in terms of the extent of melting point depression, and then also the rheological characteristics of the formulation that we're developing. Essentially, the more melting point depression that you see, or the lower melting temperature that you have, generally the easier it's going to be to leverage hot melt extrusion. Similarly, the lower the viscosity, the easier it's going to be to create a single phase system. And so these allow us to make some early assessments around viability of the platform. Although less frequently seen, depending on the quench kinetics, you can actually potentially drive to a state where you would see recrystallization occurring during the cooling event. I think in all the years that I've been doing melt extrusion and all the products that I've seen across the different companies, I've only encountered this one time. It is very visually cool to see as the extrude comes out and then recrystallizes, but not frequently encountered as a failure mode in amorphous dispersion products based on my experience. And so part of setting the control strategy comes back to how do we identify the appropriate process parameters for manufacturing the solid dispersion. And at Merck, we had done some work looking at characterizing internal temperature within the extruder and then relating that back to the phase diagram. And what you see is that if you go and you do your melting point depression studies and then you kind of relate it back to the process section set points, you'll see a deviation, as you can see in the phase diagram on your right. What you're going to see, though, when you actually open the extruder, and in this case we used an IR camera to measure local temperature, is that there's actually a much better correlation, suggesting that actually there is a strong dependency of that internal temperature in terms of being able to generate your amorphous system. And it also goes to really illustrate the fact that you will have local temperature elevation during the extrusion process internal within that system. Now we can pivot over to spray drying, because similarly with this technology, very complex, it's going to lock in key attributes, and we need to make sure that they're controlled as part of how we're developing our drug product and our amorphous dispersion. So the spray drying process, essentially we atomize droplets, rapidly evaporate off the solvent. That droplet formation process is actually quite complex, happening over very rapid time scales, but you will follow a droplet drying trajectory proceeding through the phase diagram on the right that you can see. And so you go from a multi-component system to then a uniform homogeneous spray-dried particle, and that rate at which you transit can lead to different attributes within your spray-dried dispersion particle. If you characterize with techniques like XPS, you can actually see heterogeneity within an individual particle. You also get a complex microstructure where, depending on the rate of drying, you can get spherical particles, you can get raisin-like particles. If you were to cross-section them, you can actually see internal structure there as well. So a highly complex process that needs to be very well controlled. And what we see is that actually, as we work through the different processing conditions and ultimately drive towards different particle attributes, they can have impact on the ability to process downstream. So depending on the space in which you're processing your spray drying operation, your particles can actually become more compressible or less compressible, and that can have a marked impact on the ability to create the final tablet formulation, particularly if you're in a high dose case where not only are you pushing drug loading in the solid dispersion, but really trying to maximize the amount of spray dried intermediate that you have within your tablet formulation. And we can also see notably subtle differences even in terms of the DSC characterization and characteristic aspects of those materials that we produce that can be leading indicators that there could be shifts in stability, right? Looking at the melting enthalpy as a function of our outlet temperature in this case, we did see differences, although they didn't manifest in any significant issues around the ability to control. It does suggest that the spray drying process carries implications that need to be understood when developing products with the technology. Now, we've made the dispersion particles, they're uniform. Our number one enemy at this point is going to be water. It's a potent plasticizer of solid dispersions, and we can see how even storage humidity plays a role in driving down the glass transition temperature as we go to higher and higher humidities. And so packaging here becomes a really critical element. Even basic things like facility environmental controls can play a key role in ensuring that you have the right overall control of your dispersion as you're going through the manufacturing process. And in fact, what we see is that those environmental conditions do impact the extent to which water is uptaken within a drug product formulation. We also have seen that depending on the drug product composition of matter, that can drive differences in the physical stability of our drug products as well. So it's not just about the stability of the dispersion, it is truly going to be about the stability of the drug product, and the excipients in the formulation, the environment that you're in, do play a role in the stability performance that you observe. And so one of the things that we will do is run what we call product characterization studies to essentially map out the risk space for crystallization in the drug product. And we want to understand that in the context of the storage temperature and the storage humidities that the product is going to see, because that allows us then to go ahead and control the facility, control the manufacturing process, drive the packaging in a way that enables us to be in a space far removed from areas where we would see catastrophic crystallization in the drug product. Now, the reality is you will see levels of crystallinity in drug products depending on where you look and how closely you look. So again, a function of scale of scrutiny. We can use direct measurements to make these assessments. Potentially you could use this as a means to justify some level of crystallinity in the drug product. Leveraging your clinical experience could also be a direction that you could go, although most companies, I think, will be risk averse, as we generally tend to be, and we don't normally introduce material knowingly with crystalline content into clinical studies. But it is another approach where if you had a challenging product where you were unable to control the packaging, the formulation, the storage conditions in a way that ensured no detectable crystallinity, you could go in this route. Another approach is to try and leverage modeling and simulation to inform on the risk. This type of approach generally for PK is more and more accepted by health authorities, so it can also present a direction that you can go. But in this particular instance, at Merck, we had a product where we were seeing low levels of crystalline content in the drug product. There was concern around the risk that carried. There was concern around the ability to control that. And when we saw low levels of crystalline content, we also saw a corresponding reduction in the dissolution rate, suggestive of a potential impact here to PK performance. And so we actually went ahead and decided to execute a deliberate clinical study to interrogate this further. And so we had developed drug product formulations containing 5% weight by weight and 10% weight by weight crystalline API in that formulation. And you can see that as we increase the amount of crystalline content, we're able to titrate the dissolution performance of the drug product. And so we went into a clinical study. We had expected to see potentially some impact. What we saw actually from this was quite interesting. When we looked at the 5% and 10% spike systems relative to a system that was fully amorphous, really no impact on crystalline content. Now, this is definitely going to be case specific depending on the attributes of your active ingredient and how your formulation is designed. But at least in this particular instance, we had been able to demonstrate clinically the ability to tolerate low levels of crystalline content without impacting bioperformance of the product. And so with that example highlighting an instance where you can go and run deliberate clinical studies to interrogate the impact of crystalline content, I did want to wrap up around how we think about control strategy. So the ability to control the initial amorphous formation in your drug product, that's really going to be dictated by how you control the process, and that starts with a foundational understanding of the engineering principles for the technologies that you're leveraging, coupled with strong material science. There is an impact of those solid dispersions coming back to the overall ability to produce the product as well. So we see in the case of spray-dried dispersions, how you process the spray-dried material can actually impact the ability to tablet that material. The stability performance is going to be heavily dependent on the moisture and the temperature, and moisture control, both in terms of the packaging selection and even the facility operation, is going to be essential to ensure success. And in instances where it is not possible to ensure a safe operating space that would be free of crystalline content, there are opportunities to leverage clinical studies to provide more flexibility. So overall, this should hopefully give a better sense of how to develop a robust control strategy for an amorphous product. And in particular, when you have challenging products, know that there are opportunities to be creative with how you're thinking about demonstrating out the risk space for the product. So with that, I wanted to just acknowledge a large number of my Merck colleagues that contributed to this. It certainly takes a village to bring forward a drug product to market, and so there was no shortage of effort around the examples that were shown here. And with that, I could go ahead and take some questions before lunch.
Thank you, Jim. Yeah, we've got time for a couple of quick questions before we break out. Anybody? Oh, yes, Shaka.
Jim, actually, it's a really nice, good talk. My question to you, when it spiked 5%, 10% crystallinity in the amorphous, it was a spray-dried or the HME?
So in the particular example, that was with a spray-dried solid dispersion. The spiking was done by micronizing the active ingredient, verifying that we had maintained the most stable form, which was also the form to which the drug, if it was to crystallize, would crystallize to. But that was added separately from the dispersion. And we also added a corresponding amount of the formulation components of the solid dispersion to essentially have, in terms of the quantitative composition, the same components, but the small fraction of API that was present in the crystalline state. And you're probably going in the direction of, well, is that actually reflective of how it behaves if it crystallizes on the dispersion? And this was something, that we debated internally, right? When you have a spray-dried particle, that crystallization event, it initially tends to happen on the surface of the particle, so you have a local concentration. To the degree that we could, we didn't see any substantial differences in terms of dissolution performance when we spiked in relative to when we formed it in situ. And so we elected to go forward with the spiking approach. But you just as easily could go ahead and stress your product to drive in situ formation. And in certain instances, that may actually be preferred depending on the failure mode for dissolution performance.
Okay, thank you.
Very nice presentation. Just a general question. You've been at this obviously for a while. What do you still see as the significant gaps in the field when it comes to developing ASDs? What do you wish existed if you could have the ideal ASD development today?
Mm-hmm. Yeah, it's a good question. I would say, within Merck, generally, amorphous dispersion technology, extrusion, spray drying, they're established technologies. So I think from that standpoint, there's a good foundational understanding of the engineering science behind the technology platforms. There's a good basis for rational formulation selection. I think in general, though, we still run into challenges around drug loading of the dispersion, and then also challenges in being able to manufacture tablet formulations that contain high levels of these types of solid dispersions. So opportunities with either material science approaches to improve the ability to increase drug loading in the dispersion, or looking at it from a process technology standpoint to enhance productivity. Those could be beneficial, but I think in large measure we do see success with the maybe one trade-off being that oftentimes these tend to be larger than their conventional counterparts.
Hi Jim, awesome presentation. Would selection of excipients, especially the crystalline ones, lactose monohydrate or even microcrystalline cellulose, would that crystallinity affect the instability of the dispersion during tableting process?
Yeah, that is a fantastic question. I think the short answer is the materials that you select in your drug product formulation do have an impact on the physical stability of that product. I don't know that I have a good appreciation as to what attributes of the materials that you select either impact favorably or negatively those elements. We have seen, particularly with film-coated systems, those can tend to be a very strong destabilizer of solid dispersions, resulting in low levels of crystallinity within those materials. We've generally thought that to be associated with the presence of certain plasticizers in those film coatings that can lead to destabilization. There could also be contributions to local content based on the hygroscopicity of materials that has an impact. Generally, what you're going to see, though, is that is always going to be kind of a case-by-case basis, and you really need to do deliberate studies to make sure that your formulation is the appropriate formulation for the product.
Wonderful. Any final questions for Jim before we break for lunch? No? All right, thanks very much, James.